types of pollution. Soil pollution is of concern because soil is an important ecological factor to support life and environment. It is a natural medium for plant growth and supplies the required nutrients for the growth of plants. Contamination of soil has occurred since prehistoric times, but the extent of such contamination has increased in recent years due to excessive use of various chemical pesticides, fertilizers, continuous emissions from industrial sources, and vehicular traffic. It's becoming sink for a wide range of pollutants, and the direct use of sewage sludge, industrial waste, and wastewater to agricultural land as source of plant nutrients has aroused serious concern as they are known to contain various toxic chemicals along with nutrients. Pollutants from the soil can reach us through various routes of exposure, which include drinking of contaminated water, taking food crops which are contaminated, and the food chain. Urban soils have no direct relationship to the food chain, but we have a direct influence on public health via suspended dust or direct contact. So we know the health hazards of pollution, so it becomes necessary to acquire better understanding of soil contamination in a comprehensive manner in order to avoid potential risks. It is important to evaluate soils of different regions for their potential risk to the biological system, as physicochemical analysis alone cannot evaluate the quantitative risk to human beings and other life forms. The genotoxicity assessment of contaminated soils using well-established bioassays has been widely documented. Inadvertent exposure of human beings to mutagens in the soil may lead to increase in genetic load, incidence of cancer through induced somatic mutations, and we know that a high degree of concordance exists between mutagenicity and carcinogenicity. Environmental biologists throughout the world, they are concerned to safeguard the human beings from such unduly exposure to mutagens. And therefore, it becomes, since we cannot rely upon, we cannot test directly on human beings. That means we need to rely upon experiments on other systems. So there are more than 100 test systems available which employ a wide variety of organisms ranging from viruses, bacteria, insects, plants, human cell cultures, and even intact mammals to evaluate the mutagenicity of chemicals and environmental mixtures. Among different bioassays, higher plant bioassays have been recommended by different international agencies to test the genotoxicity of various environmental chemicals and mixtures. The use of plants in environmental monitoring is justified by a good correlation between chromosomal aberrations in root tips and those found in animal cells. So we have a number of advantages of using plant bioassays. One of them is that they are less expensive. We require less expensive equipment and experimental material. Chromosome morphology of plants is similar to that of mammals. Response of plants to mutagens is similar to that of mammals. They have unique adaptation abilities for in situ studies. A number of plant systems have been in use for many years and are reliable. Being simple and requiring least facilities, cost and time of training the technicians is also relatively small. Among different plant bioassays, the most widely used are EMSI per root chromosomal aberration assay, Vishya-Faba chromosomal aberration assay, Tradescantia micronucleus and staminal hair mutation assay, and very recently we can also use potato disc tumor assay. Among animal assays, we have chromosomal aberration assays in bone marrow cells of rats or mice, micronucleus test in bone marrow cells of rats or mice, even sperm abnormality assay, comet assay using blood lymphocytes. Among bacterial assays, we have EMSI assay and SOS chromosome test. The literature survey has revealed that Among the different assays which are available, the EMSI chromosomal root aberration assay, EMS assay, and potato disc tumor assay have been used to estimate soil genotoxicity of various regions. EMSI chromosomal aberration assay has been validated by IPCS, WHO, and UNEP, and it provides a rapid screen for toxic effects of chemical mutagens and genotoxins. The advantages of using EMSI test are same as that of plants, because EMSI bulbs, they are available the year around. They are inexpensive. Their chromosome number and size is appropriate for such studies, and it is one of the oldest test systems for analyzing chromosomal aberrations. It was introduced by Levan in 1938 to investigate several chemical compounds, and ever since it has been widely used by researchers all over the world for estimating the genotoxicity or anti-genotoxicity of various test chemicals or agents. Say we just expose our onion root tips to any chemical or even the soil extract, and then we squash the root tips and see different kinds of chromosomal aberrations. We can also simulate the in situ studies, like we can place the onion bulbs directly in the soil samples which we want to analyze, and then we take out the root tips. Sorry, then we study the chromosomal aberrations. So this is a portion of which is of concern to us, which is a zone of cell division where we see the dividing cells in onion root tips. So this is the normal cells of onion root tips, showing the prophase, metaphase, anaphase, and telophase. And the chromosomal aberrations which we observe on exposure of onion root tips to different agents can be divided into physiological as well as classogenic aberrations. Among physiological aberrations which are attributable to spindle abnormalities, we have spindle inhibition, stickiness, delayed anaphases, laggards, and vagrants. Among classogenic aberrations which are attributable to direct chromosome breaking effect, we have chromosomal breaks, bridges, and ring chromosomes. Spindle inhibition is the random scattering of chromosomes over the cells, and it is also known as C mitosis. This is due to a complete spindle inhibition, so that the chromosomes do not come to lie on the equatorial plate. Stickiness is attributed to alteration of chromosomal proteins, resulting in change in nucleoprotein configuration. It arises from improper folding of chromosome fibers, and chromosomes, they lose their integrity and they become attached to each other by means of subchromated bridges. Failure of a chromosome or chromosomal fragment to get attached itself to a spindle fiber leads to laggards. So this laggard is a result of both classogenic as well as the physiological aberration. If the chromosome breaks and we have a chromosome fragment without a centromere, it is not going to pull to any of the poles. And if a spindle fiber fails to pull a chromosome, so then also it is not going to be pulled to the one of the poles. So we can see a lagging chromosome, and this is a lagging fragment. Both of them, they lead to formation of micronuclei. Vagrant chromosome is a weaker C mitotic effects where a chromosome moves ahead of the group. Like it is also a chance that it will not be included in the daughter nuclei, and it will result in micronucleus formation. Delayed anaphase is a configuration where two anaphasic groups of chromosomes lie close to each other near equatorial plate, resulting in, so that they are very close to each other. This indicates their low speed of movement. Partial C mitosis, which may lead to abnormal anaphases where chromosomes segregate in more than two poles, tripolar, tetrapolar, pentapolar, or multipolar. Chromatin bridges, where the chromosome stickiness and subsequent failure of the anaphasic separation leads to unequal exchanges, resulting in the formation of dicentric chromosomes, which are pulled equally to both poles. Ring chromosome, loss of telomeres and few, broken ends, they form a ring. Chromosomal breaks, this is considered to involve the DNA molecule responsible for linear continuity of the chromosome and may be due to unfinished or missed repair of DNA. So this is a review of some of the studies on use of EMSI by chromosomal aberration assay on genotoxicity analysis of soil. We have some studies from Amritsar, from our lab, where we have used road and railway track side soil samples, agricultural soil samples for genotoxicity and assessment, and we did find significant genotoxic effects in this assay. And then we have study from Kanpur, from Brazil, another study from Amritsar, then from Bulgaria, Ohio, USA, Lucknow, Nigeria, Mexico, France, Illinois, Ukraine, Ohio again, and Behrampur. So this shows some of the research papers where where the scientists have done a number of studies on soil genotoxicity using EMSI assay. The next essay, this is actually devised for studying the anti-tumor effect of plant extracts, but we have tried to use it for studying the genotoxicity of soil. So originally devised to determine the anti-tumor effects of different plant extracts, but it can also be employed to determine the tumor-inducing effect of a plant, of any agent. It's based on Agrobacterium tumefaciens infection on potato discs. Agrobacterium induces crown galls in more than 60 families of dicots and many gymnosperms. Crown gall tumor is a neoplastic disease of plants which is characterized by transformation of normal cells into autonomous tumor cells. Relevance of the crown gall tumor system to general cancer problem has been thoroughly reviewed. Agrobacterium shows similar pathogenicity as the two bacteria which cause tumors in humans, that is Bartonella henselae and Heliobacter pylori. During infection, the agrobacterium has a TA plasmid, which is tumor-inducing plasmid. A portion of the TA plasmid is transferred to plant genome, where it is integrated and expressed along with host genome. This TA plasmid codes for opines, which are formed in infected cells along with some enzymes which are necessary for the production of hormones. Agrobacterium uses opines as a source of carbon and nitrogen, whereas the hormones, they play an important role in conversion of normal cells into tumoral cells, thus promoting the tumor growth. So TA plasmid results in proliferation and inhibition of apoptosis similar to animal cancer cells. So this is crown gall, which is seen on the potato disc in vitro studies, and this is in nature on a tomato plant. This is how it is formed, and this is due to infection with this bacterium. So this is the bioassay. I hope you will not be able to see it. We just cut out the platinum potato disc by using the cork borer, taking the cylinders and then cutting the disc and place it on the agar plates, and then we pour our and the soil extract which we want to study for its potential for for inducing the tumors. So this is how we stain it. After we just we pour the agrobacterium, then the soil extract, and then we keep the plates in incubator at 27 degrees centigrade for nearly 16 to 18 days, and after 18 days we observe these crown galls. So you can see this is a bigger crown gall. We stain this potato disc with the Lugol solution, which is a solution meant to stain the starch. So potato discs are rich in starch, and the potato discs, their starch is stained black, and the tumor cells, they are white in color. So treatment of these potato discs with the agrobacterium and the soil extract has shown the increase in amount of tumors induced by the agrobacterium alone. So this is one study which we have reported to show the tumor inducing effect of soil extract from railway track sites. Then AIMS assay is also used to study the genotoxicity of soil extracts. As we all know that this depends upon the use of the bacterium Salmonella typhimurium, and it also shows a strong correlation between mutagenicity and carcinogenicity. And several strains of Salmonella typhimurium are used, which carry mutations in a gene involved in histidine biosynthesis. So these strains are auxotrophic mutants. That means they require histidine for their growth. This method tests the potential of a substance to induce reverse mutations that enable the bacterium to revert from histine auxotroph to histine prototroph. So rat liver extract is also used to simulate the effect of metabolism, because some compounds is like benzoypyrene, which are not mutagenic themselves, but they require but their metabolic products are. So S9 mix, which is taken from the rat livers, that simulates the activity of the metabolism within the body. So bacteria are spread on an agar plate with small amount of histidine. This small amount of histidine in the growth medium allows the bacteria to grow for an initial time and have the opportunity to mutate. When the histidine is depleted on only those bacteria which have mutated, they will gain the ability to produce their own histidine and will survive. And the plate is incubated for 48 hours, and mutagenicity of a substance is proportional to the number of colonies observed. So two test strains are most commonly used, and TA98, where the range of spontaneous revertants is 20 to 50, and TA100, where the range of spontaneous revertants is 180 to 240. This is the method which is used for AIMS assay, and this is how we take the Salmonella strain and we add the positive mutagen in the top agar, and then we plate it on the minimum nutrient medium without histidine, and then the agent which has induced mutations, we see more number of colonies, and this is a spontaneous reversion. So this shows just the number of colonies which are produced, and some of the studies where soil genotoxicity has been tested by employing the AIMS assay. One is from Kanpur city, another is from Brazil. Then we have from Amritsar, Aligarh, and from Ohio, USA. Still another study. These are some of the screenshots. And this is me and my students, and the ones which are blinking, they are the students who have contributed. They made a part of this study for their MSc, MPhil, or PhD thesis. Thank you very much.
The paper is open for discussion.
So Dr. Avinash Nagpal, would you please come out to the stage again, and may I request Dr. P.M. Gopinath to hand over a memento to her. That was right on time. We are moving ahead with the second presentation by Dr. Nanji Kumar. The title is Azadirachta indica, a unique mitigator of genotoxicity and gastrointestinal ulcer. Dr. Nanji Kumar is a former Pro-Vice Chancellor and Vice Chancellor of Magad University, Bodh Gaya. Dr. Kumar is currently a professor at Magad University, Bodh Gaya. He received his PhD from Ranchi University. He has made significant contributions to cytogenetics of cultivated crucifers and produced several interesting mutants of radishes. His current research areas are cytogenetics, biochemistry, and genotoxicity. He has published over 70 research articles and reviews in international and national journals, and he has edited 3 books for his credit. He is a recipient of Bharat Gaurav Award in 2010. He has the credit to represent India to participate and present his research paper at the International Conference on Environmental Mutagens held at Florence, Italy in August 2009 and at the second Asian Conference on Environmental Mutagens at Pattaya, Thailand in December 2010. Without wasting more time, let me welcome Dr. Nanji Kumar for his presentation. Please.
Thank you very much. I am also thankful to the organizer who gave me some opportunity to express my recent findings on particularly on some plant extracts, that is Azadirachta indica, usually called neem. In fact, the presentation will focus typically on the mitigation process, how a mitigation from neem is very helpful uh, in, in introducing and in eradicating diseases like peptic ulcer. First of all, I would like to mention that today's scenario of our industrialization and urbanization has impacted much more, particularly by use of several kinds of pesticides in our agricultural systems. And the pesticides, not only they have upset the ecosystem, rather create many cytogenetical changes in the living organisms. Similarly, okay, uh, I will mention here that there are various parameters which have been adopted here to assess particularly the genotoxicity, and that is a mitotic index, the chromosomal abnormalities, and uh, then the I think DNA content in the meristematic cells of Vishya fava. As far as uh, the various procedures concerned, it is, I think, usual the leaf extract, preparation of the leaf extract of neem and treatment of the meristematic cells with the pesticide solution. And usually the frequency of the cell division, that is mitotic index, chromosomal abnormalities, and usually the DNA content were recorded. Well, as regards the gastrointestinal ulcer, three different parameters were adopted: gastric secretion, ulcer index, number of ulcer per albino mice, for which the albino mice with ulcer-induced pyloric ligation were administered with ranitidine, a very usual and a common peptic ulcer drug, and as well as the neem leaf extract also in different doses. And ulcer index was calculated by the ratio of usually the total ulcerated area and total mucosal area, while the percent inhibition of the ulcer was calculated by control minus treatment by control. And the entire data were analyzed very carefully, and certainly results were very astonishing. Here is the Azadirachta indica plant, uh, the very common in North India, South India, all the way. Uh, the usual active ingredients found in Azadirachta is azadirachtin and what we call the limonoid also, and the usual whatever the genotoxicity experiments were conducted, uh, conducted at various stages of mitotic division, particularly the abnormalities like laggards, fragments, disturbed metaphase, stickiness, micronuclei, etc. And certainly, data, whatever collected, this is very interesting to say that uh, the pesticide like Roger, which induced this chromosomal aberrations significantly, although the mitotic index was reduced again significantly. And this histogram will tell very clearly that the mitotic index, of course, greatly reduced up to the level of even lesser or equal to that of the normal one. Certainly, the data found on treatment of for neem leaf extract, the chromosomal abnormalities, mitotic index, and DNA contents all and all parameters data collected. But interesting thing was that in comparison to the pesticide, chromosomal abnormalities were minimum, rather lesser in comparison to the pesticide, no doubt, and the mitotic index was also greatly reduced quantum in the presence of this leaf extract. Certainly, a synergistic effect will show. This is the same histogram representation. The synergistic data was very interesting because what we did in our experiment, that Roger, a pesticide, and neem leaf extract both were given in combination, and the data very interesting that so far as mitotic index concerned, uh, it was uh, it was almost reduced to much level up to the level of the control. So this indicated that neem leaf extract or the ingredients of neem leaf extract impacted vigorously and particularly in minimizing the effect of pesticide, either either individually, or there was somehow DNA repair system, or the damaged DNA was repaired. So these two possibilities. So the histogram will also saying the same thing, and certainly the neem leaf extract here reports to be a unique factor to minimize the damages induced by pesticides. Certainly, uh, here is the analysis of variance clearly confirms the fact that the treatments, among the treatments, there was different, that is a variation at the level of 0.01 p. So probability value at 0.01 p confirm that there is a significant difference between the two treatments. Certainly, now I'm coming towards the genesis of the ulcer development in albino mice, and here is the technique, simply dissecting the stomach and measuring the mucosa area and recording the ulcer index, and then certainly the number of ulcer. So here is also dissected a stomach having the normal mucosa, and the data on such things, that first of all, the the induction of ulcer was done in one set of experiments, and then uh, then there was combination of ranitidine, that is the chemical drug, and then the neem leaf extract combined, and then data were observed. So here also results very astonishing and promising, in the sense that gastric secretion, that is secretion of gastric acid from the mucosa of the stomach, uh, the, at the level of 0.66 in control, it was reduced to 0.33 in case of ranitidine plus neem leaf extract. Certainly, the same thing in ulcer index also, the same similar results came, and the same similar results also came in case of the number of ulcer. So this data was analyzed statistically, and it was found out that between the two treatments, two kind of treatments, probably here also neem leaf extract impacted greatly. That is influenced greatly the effect of gastric secretion, that is gastric secretion, particularly the ulcer index and number of ulcers in mice. And here also a significant variation was recorded, and that was at the level of 0.01. So this clearly indicate that the 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 gastric secretion is probably anyhow anyhow regulated or repaired, uh, done by the usual control values, or the ulcer caused. And therefore here is the clear cut data, and the the analysis of their impact or the impactness clearly shows that the combination of ranitidine and the neem leaf extract affects greatly, and it not only minimize the effect of ulcer in a stomach, that is peptic ulcer. Rather, it suppresses the effect of somehow the chemicals which are responsible, probably causing the peptic ulcer. So here also the same analysis of the data clearly confirms that between the treatments there is a significant impact on this peptic ulcer, and at 0.01 level in all the three parameters: gastric secretion, the ulcer index, and the number of ulcer. In all the three parameters there is a clear-cut, significant variation is here. Certainly, this is a simple presentation of the histogram, the same thing. And now I'm coming to here some sort of important configurations on such combined effect of the chemical drugs and the neem leaf extract. As far as genotoxicity concerned, uh, here the no doubt the chromosomal aberrations probably due to metabolic imbalance, all that, and failed to alter the DNA content. But interestingly, the combination of Roger and the neem leaf extract significantly inhibited this drastic reduction of mitotic activities, and this proved clearly, this confirms clearly that somehow neem leaf extract has a greater impact over the chemical pesticides. Another comparative results comes on the um, unique potential for the inhibition of AMI. AMI, no doubt, the combination of Ruger and neem leaf extract not only restore the mitotic activities, rather it came to the control level. So this is the uniqueness of the Ajar directa leaf extract. And as far as Grant's opinion are concerned, the plant system appears to be an excellent indicator of genotoxic effects of chemical mutagens. Therefore, the plant system must be accepted as a first tier assay for detection of possible genetic damages. And similarly, in case of the comparative studies of ranitidine and then neem leaf extract, a significant results prove that neem leaf extract has a potential, a high potential to reduce gastrointestinal ulcer, and the combination of ranitidine and neem leaf extract inhibited the ulceration, ulcer index, and number of ulcer spots significantly, probably operating with a unique genetic mechanism of repairing system. As far as the probable mode of action, although molecular studies will confirm all such things, but possibly there might be the ulcerogenesis due to certain ulcerogens like aspirin or the ibuprofen chemical drugs. But it has been well found from literature that it is Helicobacter pylori which causes probably this ulcer. But the exact cause will be found out from molecular studies. Although the non-steroid anti-inflammatory drugs are also responsible for this ulcer, peptic ulcer. So it is just presumed that the generating ROS probably causing the gastric mucosa, and as a result, the gastric acid synthesis or secretion will be caused, and that results the gastrointestinal ulcer. Possibly, I'm here concluding that neem leaf extract either helps repair the DNA responsible for genotoxicity or the ulcerogenesis, or suppresses the action of chemicals. However, the mechanism of the anti-genotoxic and anti-ulcer activities can be best revealed after a thorough molecular analysis of the bioactive compounds like azadirachtin. Certainly, our finding and our recommendation would stress upon the fact that the neem leaf extract may be used as a suitable substitute for the chemical pesticides with minimum risk of genotoxic damages, and a natural antacid for mitigation of peptic ulcer or gastrointestinal ulcer, that will be far superior in comparison to the chemical drug like ranitidine. So this is our finding, and certainly acknowledgements go to the Magad University Botany Department, Mahavir Cancer Institute, Patna, and the workers were Vishnu Shankar Sinha, Vikas Kumar Prabhat, Nilmani, and Mukesh Kumar. Thank you very much.
Thank you, Professor Nanji Kumar, for a wonderful presentation. The topic is open for discussion. I would like to open the discussion with a question. Professor, it was a very interesting observation that the ranitidine and the neem leaf extract 80 and 160 were able to reduce the gastric juice secretion almost to the same extent. And I have seen a very few very elderly people consuming a few neem leaves every morning.
Yes.
So can you comment on the dosage that they need to take about when you say that NLE 80 and 160? So how much raw leaves it will amount to approximately?
In our results, I think 80 mg per kg of mice. So 80 mg has better results. So as far as chewing in the morning, so far as concerned, in my opinion, at least 5 weeks enough for a normal human to take it in the morning. So this dose has been supposed to be with better results, with preventive as well as the curative.
Excellent, Professor. So questions from audience, please.
Okay.
I think at higher concentrations, I have seen some reports that this may be neurotoxic also.
Neuro?
Neurotoxic. Have you seen such reports? Can you
No.
comment on that?
No,
Higher concentrations of this extract may be neurotoxic. I have seen some reports.
but we have not seen it. We shall see it in the next series of experiments. Okay.
That means professor has not gone to the concentration that may cause neurotoxicity. Probably he is playing well within the limits that is going to give good results.
So in this case, I think dose dependent will be the most important fact. So dose is important. Anything more?
Um,
Thank you, Doctor. Now, Dr. Raviraj will present a memento.
Our next speaker for the session is Dr. Satwinderjit Kaur. The topic is cancer preventive potential of Lawsonia enormis. Dr. Satwinderjit Kaur is currently professor and head of the Department of Botanical and Environmental Studies, Guru Nanak Dev University, Amritsar. Dr. Kaur has received her PhD from Guru Nanak University. Her areas of research, she has been very active in the field of anti-mutagenesis and anti-carcinogenesis of natural plant products and environmental toxicology. She has over 70 publications in the field for her credit. She has been awarded with Professor P.N. Mehra Memorial Young Scientist Award, Boy Scout Fellowship from DST, New Delhi, and DBT Crest Fellowship from DBT, New Delhi. So without spending more time, let me welcome Doctor for the presentation. Thank you.
Thank you very much, Chairman. And at the outside, I would like to thank the organizer, Professor K. Satyamurthy, for inviting me to deliver my talk, my research findings to this August gathering. Thank you very much. So in this presentation, I would like, my topic is cancer preventive potential of Lawsonia enormis. This Lawsonia as such, so our lab is working on different medicinal plants, and we have started from uh, finding out the anti-mutagenic potential of various medicinal plants, and then we have now moved over to cancer chemopreventive effects of various plants. So as you know, this cancer chemoprevention is this involves the prevention, delay, or reversal of the process of carcinogenesis through the utilization of either the natural products, or it may be synthetic or other biological substances. So as there are various mechanisms that work on for the various cancer preventive effects shown by various uh, uh, these chemopreventive agents. So the plant extracts as such, they have uh, to some extent, although we say that in plant extent, what is the actual constant which is affecting, which is showing the effect, but still plant extracts, though they are the mixture, but they have, since they hit multiple targets, and they also show various pleiotropic effects. So, and the compounds resulting from the plant products, these are well known. These are all the camptothecin, podophyllitoxin, and this paclitaxel. They are the plant-derived drugs, and they have influenced the anti-cancer research. So these chemopreventive agents, they can interfere in any stage of the carcinogenesis. So these are various mechanisms, the anti-mutagenic mechanisms, and these are the various anti-proliferative mechanisms. As I told you that we have in our lab, we have worked on different medicinal plants as terminally Arjuna on the bark, which is also cardio protective. And we have, and then Rubia cordifolia, the rhizomes, Glycera glabra, And from these plants, we have been able to isolate various phenolic compounds, and they have shown the anti-mutagenic effect. Ale marmelos. From ale marmelos, we have also been able to isolate certain resinosteroids, which have proved to be and has shown anti-genotoxic effects. Uh, Centella asiatica. Then recently we have worked, one of my student has worked on Anthocephalus kadamba, and we have been able to again isolate various alkaloids, especially, which are actively, they have shown the anti-genotoxic effects. Now, a few of my, this we have started the pteridophytes. Recently, one of my student is working on Pteris vittata, then Cassia fistula, Butea monosperma. So now at the present study, I'm just focusing on one of the plants which we had recently initiated, commonly the henna plant. So this is monotypic genus. It is having one species. This is Lawsonia enormis, and belonging to family Lythraceae. So this is how we have prepared various extracts and fractions. Uh, and So this is the, we have initially uh, taken out the methanol extract, and uh, the then it was the various fractions. We isolated various non-polar fractions in the order of increasing polarity of the various solvents, as hexane, followed by the chloroform, and then we have ethyl acetate, then and butanol, resulting in these fractions. And separately we isolated also, we made a water extract. So then we went on to seeing the various, the phenol, we estimated the phenolic and flavonoid content, and we saw that this is the mother extract. The methanol is having all the, it's rich in these three, methanolic, ethyl acetate, and the butanol fractions. They were rich in phenolic compounds. Then most of the culprit is the ROS. So we went on to uh, see the antioxidant activity of the various extracts that we fractionated, and based on the set hat based single electron transfer and hydrogen atom transfer based assays are resulting in the end results. So these are various assays that we FRAP assay, reducing power assay, BTS, DPPH, and deoxyribose degradation assay. And we also saw some biologically important assays, the superoxide radical scavenging, DNA protective assay, and then lipid peroxidation. So then looking at the IC50 values of the various extracts, we were able to pinpoint. So these some highlights are when the IC50 values is below 100 microgram per ml. So methanol, ethyl acetate, and butanol really worked well. They showed good antioxidant activity. So then we went on to actually see what all our constituents are present in the various extracts that are showing effects. And then we, by with the way of UHPLC, we tried to see the various constituents, and we saw that the these ethyl acetate and butanol, they are rich in catechin, chlorogenic acid, and elagic acid. And this is, of course, the mother extract, and some it's camphorol also is there. This is the plasmid nicking assay, where using PBr 322, and then we saw how the extracts, they protected the DNA from the hydroxyl lines using Fenton's reagent. And so these are the different extracts. And sorry, this is again the same. So then we saw that this concluding that these ellagic acid, chlorogenic, gallic, camphorol, and catechine, they are probably so these are the DNA protect showing deep DNA protective efficacy, and this antioxidant efficacy is due to these chemical constituents found in the various extracts. So this is one of the publications reporting the polyphenols with showing the antioxidant, anti-genotoxic, and some anti-proliferative activity. So then we took one of the extracts, the butanol fraction, and we tried to see how it protected from the hepatic damage caused by the 2-acetylaminofluorine. So this was the designed experiment. So we have the group 1 as control, and then we have the vehicle control. The third group is the positive control, and we took 3 concentrations of the butanol extract, and then we saw it on the effect of various parameters. So this SGOT, this is the red line shows the uh, enhanced uh, uh, by the 2-acetylaminofluorine, and the treatment by the butanol various concentrations 100, 200, and 400 milligram per kg body weight. So they reduce their levels, showing the hepatic these elevation of the hepatic toxicity effects. And then we also saw it on the T-bars. So they are again pointing towards the hepatoprotective effect. So this is the during seeing at the histological studies. So this was the different. So this is the 2AF treated group, and this HAI score was 6/18, and then with the increase in contraction, this is coming to the normal level. So these are the various sections. This is the control group, vehicle group, and then this is the positive group, and these are the different concentration of the butanol extract, showing the hepatoprotective effect. So this is the recent publication coming in the BMC, complementary and alternative medicine. It's just come out in 2017 publication. So this reporting the hepatoprotective potential of Lawsonian enormous leaves against 2AF in male Wister rats. So this the leaf powder, so it is inhibiting the ROS, and we have seen in vivo, not only in vitro assays, we have demonstrated that it is showing protective effect in against ROS, and also in vivo uh, the by this 2AF induced hepatic damage. So then we went on to see the anti-proliferative activity also in the various extracts fractions of this plant. So we did some the cytotoxicity assays, cell viability, MTT assay, clonogenic assay, and then also then we went on to see that if it works by it has the apoptosis inducing activity using these various techniques, confocal. Then we used cell cycle analysis using flow cytometry, scanning electron microscopy, and measuring the MMP, that is mitochondrial membrane potential, using flow cytometry. So when we saw this using MTT assay, we worked on these 5 cell lines, and these had these are the GI50 values that we got, and this hexane extract and the chloroform extract to some extent, although it was not very active in all the cell lines, but throughout it showed some effect in various cell lines. And then we just went on for the various mechanisms by which it may work using HeLa cell line. So this is the colony formation inhibition in HeLa cell lines cells shown by the various concentrations of hexane and the chloroform extract of Lawsonia. So this is the morphology. This is control cells, and then the different concentrations of the hexane extract using the phase contrast inverted microscope. So this is the of the chloroform extract, the control and the different concentrations added on of the chloroform fraction of Lawsonia. So this is, although it sets 40x is not clear. So this is some nuclear changes in the nuclear morphology when we saw it in under the confocal microscope by DAPI staining. This is by the hexane extract. So then this is by chloroform extract. So this is the confocal measurements wherein it measures the percent apoptotic cells which are induced. So these are showing the increase in number of the apoptotic cells as determined in the flow cytometry, and they are in the sub G1 phase. So these are the SEM photographs of this is the HeLa cell and treated by hexane extract and then by the chloroform fraction of Lawsonia. So this is the measurement of MMP potential in HeLa cells with treatment of hexane and the chloroform fraction. So this is one of the publication coming out depicting the antiproliferative apoptosis inducing effects of these non-polar fraction, that is hexane and chloroform from Lawsonia enormis in HeLa cancer cells. So probably to conclude, the studies that we have done so far is that this Lawsonia, it's harboring the various polyphenols, and it is having antioxidant activity, and which is inhibiting the ROS. And in vivo studies, we've seen that so all these factors they are being inhibited, which are these are indicators of the hepatic damage which has been reduced. And then also it is showing cytotoxic and apoptosis inducing effects which are studied in HeLa cells. And then so various the apoptosis induction is by ROS accumulation and decrease in MMP. Then we also did, which the results have not been shown here in COMET assay, and there is increase in sub G1 population, that is apoptotic cell population. So thank you very much for your kind attention.
Thank you, Professor Kaur, for the wonderful presentation. The topic is open for discussion. Questions from audience, please.
You said that you studied the protection against OH radicals. What was you studied that it protects against the OH radicals? What was the system used for this study? Not you have not used radiation to induce the OH radicals.
Any further questions? I would like to say that the 40th annual meeting was held in Amritsar last year, and Dr. Kaur organized this meeting wonderfully and very successfully. That's what I have been informed by Professor Gopinath. Thanks, madam. And it was a very good work starting from the leaf, starting from the extract, doing a lot of the in vitro assays and taking to preclinical level and figuring out which extract is better. Probably the foundation is laid for further clinical trial by some companies. Thanks, doctor. And I will request Dr. P.M. Gopinath to offer a memento as a token of our appreciation. Before we proceed ahead, there is an announcement.
There's a small announcement. I request all the participants of Young Scientist Award to report at the registration desk, please. Thank you.
Oh, correct. So let us move on to the next speaker, Dr. Pooja Sharma. The title of the topic is Peroxidative Metabolism After Beta-2 Agonist Phenoltirol on Swiss albino mice. Dr. Pooja Sharma is currently associated with Himachal Pradesh University, Shimla. Her area of interest is the broader toxicology. She has published over 12 publications to her credit, and she had been awarded with the Junior Scientist Award by NESA. Dr. Pooja Sharma, you have 15 minutes. Please start. Thank you.
Thank you so much, sir. Very welcome to all of you. I'm going to present and my topic entitled with Peroxidative Metabolism after beta-2 agonist phenoltirol on Swiss albino mice. The organism I have chosen for this work was albino mice, and the organ was small intestine having all the three parts: duodenum, jejunum, and ileum. Phenoltirol. It is a drug which is a beta agonist which relaxes the smooth muscles. It is clinically used as a bronchodilator. It relaxes the pregnant uterus. When peroxidative metabolism occurs, it occurs by mono and 1,3-dihydroxybenzene moiety. Oxidation causes their transformation through the intramolecular cyclization by the addition of their amino nitrogen to the aromatic ring. Phenoltirol acts as substrate for peroxidases and resulting product that differ in structural as well as morphological properties. Phenoltirol actually exists in the form of hydrobromide. Bromide ion interferes with the enzymatic oxidation of the drug. Now, what is oxidative stress? It is the disturbances in the normal redox state of a cell. Production of peroxide and the free radicals takes place. It damages the cell, including protein, lipids, and the DNA. Damage is actually caused by reactive oxygen species, which act as cellular messenger in the redox signaling. It results in programmed cell death and traumatic cell death, that is apoptosis and necrosis. ROS can be beneficial as they are used by immune system to kill the pathogens. Short-term oxidative stresses may be important in prevention of aging by induction of a process called mitohormesis. Mitohormesis is a process in which when any substance is taken in small concentration that might be beneficial for that, it increases the life expectancy by increasing the ROS that provide resistance to the cell against the oxidative stress. This is the schematic diagram which show the induction of oxidative stress and its pathophysiological conditions. The induction factors could be any drug or any infection, smoke, alcohol that produces the ROS. Cellular responses are generated by the oxidative damage of DNA, protein, and lipid peroxidation that altered the signal transduction, and pathological conditions are generated. These are epithelial injury in the small intestine, endothelial injury, and inflammation of the cells that results in ulceration, fibrosis, and ischemia even in carcinogenesis. Next is the structure of this drug. If you see the structure of this drug exerts its effect via two receptors, beta receptors. It has the anionic site to which bind with ammonium group, hydrogen bonding area which bind with the phenol group, and third one is for the aromatic ring. Phenetrol was manufactured by Boehringer Ingelheim. Beta 1 receptors are present in the heart and the kidney. Beta 2 receptors, they are usually present in the vascular smooth muscles, small intestine, uterus, liver, bronchi, and the skeletal muscles. Beta 1 stimulation causes various side effects, and beta 2 stimulation causes labor inhibition without any side effect. Phenetrol is beta 2 selective. Therefore, it is associated with fewer beta 1 side effects like tachycardia as well as hypotension. Beta 2 agonist that leads to improvement in the oxygenation and was sold under the name Barotec in New Zealand. Phenetrol show anabolic actions in the skeletal muscle, produces changes in the mass, causes hypertrophy. Like phenetrol, other sympathomimetic substances like clenbuterol, albuterol also activate the beta receptors in vivo and enhance the growth of the tissues. Now the mechanism of action of this drug. There are two types of receptors, beta receptors and alpha receptors. Beta receptors are further of two types, beta 1 and beta 2 receptor. They are antagonists of both. Beta 1 show excitatory responses and beta 2 inhibitory responses. The receptor, beta 1 receptor has specificity. It has a regulator protein which interact with the quinine nucleotide sensitive regulator proteins. And regulator protein, they activate the adenyl cyclase and convert ATP into CAMP and modulate the beta adrenergic responses. Next, for my research, I have taken the level of the SOD because it is antioxidant enzyme on all the three parts of the small intestine, that is duodenum, jejunum, and ileum. Then I have noticed the level of MDA after drug administration in all the three parts and the concentration of the protein after oxidative stress. Material and methods. The experiment was performed by taking the two groups. First one is the control group and second one is drug treated. I have taken 1.4 mg per kg of body weight for 28 days. The estimation was done for 7, 14, 21 and 28 days on all the three parts of small intestine. SOD activity was measured by Mishra and Friedovich method and MDA and was by the method of Dhinsa et al. Proteins were estimated by the method of Fleury et al. These are the observation after doing the experiment. ROS are generated after oxidative stress. Therefore, ROS that oxidizes the proteins and cross-linking of the proteins takes place. After this, accumulation of the proteins takes place and carbonyl group get attached on the side chains of the protein that leads to the increase in the content of the proteins. Significant increase was noticed at 28 days. The protein concentration increases after the treatment of the drug. Next one is the lipid peroxidation. As you all know that oxidative stress increases the lipid content in the cell. I have measured in by the TBA method in all the three parts. The ROS that degrade the actually lipid bilayer and inactivates the enzymes. Therefore, the concentration of the lipids also increases from 7, 14, 21 and 28 days. These are the histograms which show the activity of the superoxide dismutase. Increase in the concentration because it is the antioxidant enzyme. After this drug, when the oxidative stress occurs, increase in the level of the SOD takes place from 7 to 28 days. Now these are the histological studies of all the three parts. First, I have started with normal duodenum. Normal duodenum showed normal level of the adventitia, Brunner's gland, then villi are normal. Submucosa level is normal, but after the phenetrol administration, you can see the increase in the muscularis propria, Goblet cells increases, Brunner glands also increases. You can see the increase in the length of the villi. After 14 days of the drug treatment, we have seen disrupted enterocyte cells. Goblet cells get inflamed. Plasma cells concentration increases in the lamina propria. Plasma cells actually produces antibodies which are of two types, IgA and IgM antibody. IgA antibody is produced in huge concentration and it provides a kind of immunity to the mucosa so that the microbes could not get attached with it. After the 21 days of drug treatment, you can see the necrosis of the lamina propria, and 28 days of drug treatment, duodenum show atrophy and degenerative changes. The lamina propria that show exudation and villi architecture get completely lost. This is the normal jejunum. We show lamina propria in a proper condition and the crypts. After drug treatment, the length of the villi get increases, goblet cells increases, inflammation of the goblet cells takes place, Brunner's gland get modified, and at 28 days of drug treatment, sloughing of the nuded villi takes place. This is the section of normal ileum. After drug treatment, inflamed epithelium you can easily see with the loss of crypt architecture. This section is for the last stage, 28 days after drug treatment. We show increased lymphocyte, increase in the plasma cells, and increase in eosinophil in the lamina propria. Eosinophil, actually, they release the hydrolytic enzyme which act on the worms like pinworm, tapeworm. If they are in the intestine, they can, by releasing the hydrolytic enzyme, they can engulf it. They show the phagocytosis. Phenetrol is administered in the mice in order to evaluate the beta-adrenergic and toxicological action in terms of lipid peroxidation and SOD. Hypertrophy was noticed in all the parts and increased in the MDA content, increase in protein content. Oxidative stress causes pathophysiology like gastrointestinal cancers, inflammatory bowel diseases, and Crohn's diseases that in inflammation from mouth to anus. Now, to assess the criticism that beta agonists may pose significant risk if used regularly over a period of time, future studies should include long-term trials on large population sample with hospitalization and other healthcare utilization measure outcome variables. Many cell type within the mucosa of gastrointestinal tract produces the reactive oxygen species as a part of normal physiology. Yet the mucosa also is a target of various oxidants that can lead to pathological conditions. Oxidative stress markers a substantial contribution to the pathogenesis of GI mucosal disease, and despite recent progress, mechanism of ROS-mediated GI diseases are not well established. Understanding the cellular and molecular mechanism, including altered signaling of ROS and critical developing future therapies for GI diseases mediated by oxidative stress, future treatment strategies for these important mucosal GI diseases lie in a better understanding for free radical biology of the gastrointestinal mucosa. Thank you so much. Thank you for the patience.
Thanks, Dr. Pooja, for a very clear presentation. The topic is open for discussion. It appears everybody has understood everything. So let us move on.
Now Dr. Raviraj will present a momentate to her.
You heard it, sir. Let us move on with the next presentation by Dr. Sweetie Raju Kanat. The topic is bioactive properties of mango peel and its utilization in food packaging. Dr. Sweetie is a scientific officer at BRC, Mumbai, and her areas of research interest are food additives, natural antioxidants, food packaging from biopolymers, radiation processing of food. She has published over 25 publications in peer-reviewed national and international journals. Dr. Sweetie, please.
Thank you, Chairman. Good afternoon, ladies and gentlemen. At the onset, I have to tell you that I'm a food technologist and I'm addressing an audience in which I think there are experts in toxicologists, molecular biologists, and students who are aiming to become like them. So, I will be sincerely trying to tell you how relevant is my work in this forum. It's more of an application based study. So, the topic is bioactive properties of mango peel extract and its utilization in food packaging. Yeah. So in today's world, we know that we are consuming more and more of processed food. Consumer demands ready-to-eat food, ready-to-cook food because we don't have much time. So any food that is processed will have a lot of food additives. Even earlier times, we know that our grandmothers prepared, processed food like achars and papads, but they were using conventional additives like salt, sugar, vinegar, or to prevent oxidative reactions, they were using spices. But now, with the plethora of processed food that is available, we cannot limit ourselves to these traditional food additives. So, a lot of food additives like they have to be added to the food when it is processed, because food additives basically they have got several functions. The first one is that they preserve food. That by two mechanisms. They have got antimicrobial activity as well as antioxidant activity. So the antibacterial activity earlier, as I said, it was by using salt, sugar, vinegar, but today we use sodium benzoate, sulfates, and many other compounds. And to have prevent lipid peroxidation, earlier we used to use spices. Now we use lots of synthetic antioxidants like BHA, that is butylated hydroxyanisole, butylated hydroxytoluene, propyl gallate, and many more. Now, many of these antibacterials and antioxidants, which are synthetic, though they are approved, but the consumer has the fear of consuming it because many of them are in long term have been proven to be carcinogenic or mutagenic. They also, food additives have to be added because they enhance the taste, the texture, and the appearance. So earlier, as I said, we were using for flavor enhancement spices, but now you have got, all of us love Chinese food, right? But Chinese food, there is a lot of monosodium glutamate. We also like processed food like sausages, salamis, which contain a lot of sodium nitrite. Then, to improve the texture and appearance, you've got a lot of stabilizers and thickeners. For example, in ice creams, bakery products, these are added. You also have food colors, which improve the appearance of the food. But many of these food colors, they are synthetic food colors, and they are very toxic or mutagenic. Also, your potassium bromate. Recent years, you must have read in the newspaper, there was a lot of controversy on this potassium bromate, which is added to bread. Saccharine, that all of you know, it's a sweetener, artificial sweetener, aspartame. All these have been shown to be mutagenic or carcinogenic. Now, to improve the nutritional value of the food, like many of us take cornflakes now in the morning, so that to improve its nutritional quality, vitamins or fiber has been added. But many of these vitamins are chemically synthesized. So the consumer is very scared about how safe it is. So the solution for this is to use natural additives from plant sources. Now, processed food what happens in processed food, it has to be packaged. Most of the processed food is packaged. And we know that packaging, mostly what we are using is LDP, HDP, PET, all your PET bottles, thermocols. These are all not environmental friendly, and they are creating a big burden on our environment. You find our sewages are choked, Our oceans are filled with plastic. Even animals, our animals like cow and all, if you see in the intestine, now you get plastic. Instead of, consuming grass, they are eating a lot of these because they cannot differentiate. So about 175 potentially hazardous substances, they have been isolated from these packaging materials, especially bisphenol A. This is used as a plasticizer, but we know that it is a toxic compound. Phthalates, melamine plates we use, but these are also toxic. Also parabens. So there are many of these additives and plasticizers that are used in plastic, which is not very safe, and plus it creates an environmental problem. So my solution is that we could have active packaging. Active because in addition to packaging, they are going to give you bioactive properties. So it is an active packaging, and you're using biopolymers instead of the petroleum-based polymers. Now, mango needs no introduction. It is the king of fruits in our country. It is our national fruit. So, summers we really look forward because that is one positive thing. We can eat lots of mangoes. So, it's a climatic food, and India is the largest producer of mangoes. Almost 50% of the mangoes that is produced is from India. India also exports a lot of mangoes. Earlier, we were not able to export to America because of quarantine reasons. Now, recently, I think from 2007, we are exporting radiation processed mangoes to America because this radiation does two things. It increases the ripening period and also it delays ripening, sorry. And secondly, the seed weevil which is there. So for quarantine purpose, we are using radiation process. So Indians in US, especially the Gujarati community over there, they are enjoying radiation process, radiation treated Alfonso mangoes. So in India, we have got about 1,000 varieties, or 1,000 or more varieties of varieties. You will see each state has got at least 10 to 15 varieties, different varieties of mangoes. And the leading states are UP, Maharashtra, Karnataka, Andhra Pradesh. And just to name a few, Alfonso, obviously, those who are from Maharashtra will vouch for it that that is considered to be the king of mangoes. Uh, Badami, that is Begumpalli, that comes from earlier Andhra Pradesh. Now I'm not sure whether it is Seemadhara or Andhra Pradesh, one of those. Then you have got Langra coming from UP. You have got Kesar coming from Gujarat. Now we produce a lot, but we are not able to export it or consume it fully. So a lot of mango is being processed in the form of juice, in the form of nectar, in the form of what you call pulp. Then you have many. It is being used also in ice creams and bakery products. So a lot of peel is being generated as a waste. Now this peel, it is very rich in bioactive compounds. So we could use that as a food additive. I'll just come to that. So here what we did is basically we took 4 varieties of mango: Alfonso, Badami, Kesar and Langra. We only took the peel. We got it from a local processor where they are processing mangoes, and the peel is generated as a waste. We dried it in an oven, made it into a powder form, and then we extracted it by 2 methods. One is by continuous agitation, wherein the powder, it is added to the different solvents and it is agitated for 24 hours, or we sonicated it. Sonication was just for 1 hour. So different solvents that we took is water, 100% ethanol, 70% ethanol, and 80% acetone. The reason is, especially where you're using a food additive, we would like to use a solvent that is safe. So ideally, we would like to use either water or ethanol and not methanol or any other organic solvents. But here we took acetone because these peels, they are very rich in carotenoids. So as you all know, we wouldn't be able to extract them in water. Now we know that basically all the bioactive compounds that are present in plants, they are basically phenolics, which are secondary metabolites produced by the plants. And so we quantitated the total phenolic content. Here you can see for the different, sorry, for the different varieties of mangoes and also for the different extraction methods, we found that the Langra peel extract that was extracted in acetone, 80% acetone, that had the highest phenolic content. Even in Kesar, the phenolic content was pretty high. Comparatively, in Badami and Alfonso, it was comparatively less. Now we know that flavonoids, they are the major class of phenolics, which is responsible for the antioxidant activity. About around 4,000 flavonoid compounds are known. Here we have not tried to isolate the flavonoids. This is the total flavonoid content. And here again we saw that the 80% acetone extract of langda peel, which was extracted by sonication, had the highest flavonoid content. And even in kesar it was good. And even the 70% ethanol extract also gave fairly good flavonoid content. Coming to tannins, this is another class of phenolics. Here, in case of langda, we saw that the tannin content was less as compared to the others. Now, tannins, it is like a double-edged sword. Like one way they say that tannins, they are anti-nutritional factors because they bind to the proteins, and so the absorption is reduced. So it is not very good. But off late, there's a lot of research. I mean, you all are experts. You must be knowing better than that, better than me, that it can be used as an anti-cancer drug because it prevents. It precipitates the proteins. So here the tannin content in case of langda was less. Now, as I told, the color of the mango is due to the carotenoids present. So we saw the carotenoid content here. Langda had it had a good amount, but in kesar the carotenoid content was higher. You will those who know kesar, you know it's more yellow in color as compared to langda, and so we got a higher carotenoid content in that. We did a lot of in vitro assays, antioxidant assays to see the antioxidant activity. Here we have what I have tabulated is the IC50 value. So obviously the lower the IC50 value, better is the antioxidant. And here we saw that langda again, sonicated, it is having the best antioxidant activity. Also by superoxide, these are the main raws which are responsible. So we have seen how well this extract can do. So here again we see that in case of langda, it is better antioxidant. Iron chelation, because we know that iron is a pro-oxidant, and by Fenton or Haber-Weiss reaction it can lead to peroxidation. So here again the langda was having a good activity. Same way for beta carotene assay. The antibacterial assay again showed us that in addition to antioxidant activity, this mango peel extract also had good antioxidant activity. We saw it against both gram-positive and gram-negative organisms. Now what we did is that we used this mango peel as a functional component to prepare film. The film was prepared with polyvinyl alcohol, which is a synthetic polymer, but it is degradable and it is biocompatible. So that's why we used it. Cyclodextrin, it's a polysaccharide, but it has got hydrophobic ring inside. So these carotenoids can bind to it. So that's the reason we use cyclodextrin. Gelatin, because it improves the permeability. And we just saw the characteristics, and we saw that the film which was prepared with langda peel extract, 70% ethanol and acetone, it did not change the characteristics too much. In fact, the tensile strength was improved. It had some color because of the mango peel. It also gave good UV protection. So any food that is packed in this packaging material, it will prevent lipid peroxidation due to UV. And it had good antioxidant release. The antioxidant which was present in the packaging film, that was released, and so it could prevent lipid peroxidation in the food that was packed in that. This is the antibacterial activity of the film. Again, here we see that it is not lost when we are incorporating it in the film. It still retains its antibacterial activity. Now we actually pack this chicken in this packet in this polymer that we have developed in packaging film. This is how it looks. This is the first one is the one control film in which there is no langda peel, and the second one is having the acetone extract, and the third one is having the ethanol extract. So we saw its shelf life. That is, we packed chicken in this packaging material, and it was stored at cold temperature, and we were periodically seeing its microbiological analysis, and we saw that even after 12 days, it was microbiologically safe, whereas the control spoiled in less than 3 days. We also saw the lipid peroxidation, and we saw that meat which was packed in this langda peel extract, it could minimize lipid peroxidation in chicken. So to conclude, mango peel, it is very rich in bioactive compounds, and of the four varieties that we studied, we found that langra peel was the best, and we could make an active composite film using PVA, cyclodextrin, gelatin, and langra peel extract. And the bioactivities properties of the langra peel was retained in the film. It had UV blocking mechanism, mechanical properties were good. It in fact improved. Shelf life of the chicken meat was extended by 10 days because control spoiled in 3 days, whereas this we could keep for 12 days. And 70% and 80% acetone extract both were effective, but where we are, if we have a choice of using it for food, then it is better to use the 70% ethanol extract. So this developed film has a potential to be used as an active film for food packaging and thus reduce the burden of petroleum-based food packaging films. Thank you.
Thank you very much, Dr. Sweety. It was a very good presentation, a kind of new concept. How do we develop peels? How do we develop films using these mango peels? The topic is open for discussion.
Sweety, congratulations. Have you applied for patent? You should try to apply for a patent if you didn't know, that's first thing. And you should also have the, what is the life of this packaging material?
Yeah, we are doing long-term studies.
Yeah, so that data also should be, because that will be very important, particularly under different conditions, temperature, heat, etc. But ask your seniors to apply for the patent.
It was a very good suggestion by Professor Chauhan, because otherwise someone else may do this and they may apply for the patent much earlier. Yeah, and also doing the stability studies as told as per the ICH guidelines and at different humidity and temperature. Yeah.
Excuse me, ma'am. Okay, so you have tried out with chicken. So did it affect the flavor in any way, or when you are?
We did the sensory. It is not effective because we are using only just 1% of the extract.
Okay. And there's I had read about an article where in Andhra Pradesh, a meat seller, he was packing the meat in some leaf, and many people were saying that the leaf not only it had like similar kind of properties. The meat would stay fresh for a longer time, and they were saying that it improved the flavor. So that's why I just wanted to ask if there's any advantage this will give over the other one.
In meat, at least, you wouldn't want to have a mango flavor. So in that case, that is not something which is really desirable. Maybe in other products, yes. But in meat, at least I wouldn't like to have a mango flavor in meat.
Yes, yes.
May I request Dr. P.M. Gopinath to offer the memento as a token of appreciation. Let us move on to the last but one presentation by Miss Shuchisnigdha Datta. Shuchisnigdha Datta. The title of the topic is Role of Black Tea on NRF2 KEAP1 signaling in A549 lung cancer cells. She is a junior research fellow pursuing PhD at Chittaranjan National Cancer Center, and her area of interest is cancer therapeutics. May I request Ms. Shujisnigdha Datta to come over to the dais.
A very good morning to all the delegates, my respected chairpersons, and all my beloved audience. I'd like to thank the organizing committee for providing me this excellent opportunity to present my work, that is role of black tea on NRF2 KEAP1 signaling in A549 lung cancer cells. Pulmonary carcinoma is the most common cancer in men. Is the most common cancer in men globally as well as in India. And with a death rate of 1 out of 4, it is the leading cause of cancer death both in men and in women. Though there are standard treatment protocols widely available like lobectomy, then chemotherapy, radiotherapy or combinations, the 5-year survivability rate is very poor. That is only 10 to 15%. So what makes this cancer the deadliest? It has been found that the inert resistance from the classical cytotoxic chemotherapeutic drugs is the reason behind this. And studies also say that the dysregulation of NRF2 pathway is the ultimate player behind this stress resistance in various cancers, including lung. This is the molecular structure and mechanism of function of NRF2, and it's which is under tight regulation of KIP1, which acts as a molecular switch. In otherwise unstressed condition, KIP1 through its DGF domain binds to the DLG and ETGF domain of NRF2, which acts like a substrate adapter for the Cul3 E3 based ubiquitin ligase complex for its 26S proteasomal degradation. But if there is any stress or chemical insult, it can KIP1 can sense it through the thiol modification of its cysteine code. As a result, the NRF2 is liberated, stable, so now free to translocate into the nucleus, where it dimerizes with small MAF and binds to the antioxidant response element. As a result, a robust induction in the battery of cytoreactive genes occur. NRF2 plays two drastically opposite role into different situation. In normal cell, it acts like a tumor suppressor by providing chemo prevention. But the same NRF2, when it is constitutively active in cancer cell, it acts like an oncogene by favoring the cancer cells with survival and growth advantage. There are several layers of regulation like gain of function mutation in NRF2 itself or in other oncogenes like RAS, RAF, MEK. Then loss of function mutation in the inhibitory machinery like KIP1 and Cul3, and there are miRNA mediated suppression too. Then loss of homozygosity, DNA hypermethylation, and histone deacetylation. The cumulative effect of all these changes keeps the NRF2 constitutively present inside the nucleus. As a result, it protects the cancer cell against all kind of stress acting on it, making it more and more tumorous. Here comes the role of polyphenols into action because they are excellent candidates for chemo prevention, and black tea is one among them. Now you will ask why black tea? I will say why not. After all, worldwide production and consumption wise, it far exceeds that of all other beverages. Moreover, the oxidation polymerization products of black tea, the theaflavin and thiorubigenes, possess antioxidant properties equal to their green tea precursors. So having a potent pro-apoptotic and cytotoxic effect, it also shows excellent lung protective effect. So, to address one of the most challenging health issue worldwide, we have selected the most consumed beverage consumed by 2/3 population of the world. Here we have designed our study in this way. In our study model A549, which is a lung adenocarcinoma cell line containing a KIP1 mutation. Here, under the feeble control of mutated KIP1 and under the excessive oxidative stress, NRF2 level is naturally active constitutively. As a result, its downstream molecules like the xenobiotic detoxifying or metabolizing enzymes, antioxidant, and drug efflux pump or the drug transporter are also shooted up very high. So in this context, we wanted to study the effect of black tea, which is a known NRF2 activator. We have to keep in mind the effect of black tea in the NRF2 KIP1 pathway of our A549 cell. First, we have done the time dose dependent cytotoxic study and have found that 0.46 microgram ml of black tea after 6 hours of treatment have shown the most potent cytotoxic effect by reducing the cell viability to 39.44%. Then we want to check its antioxidant property, and from this overlay graph, the leftward movement, we can very easily say that this particular dose was significantly decreasing the ROS generation. it reduced the lipid peroxidation level too. Then we directly went to the signaling pathway, and from the whole cytosolic and nuclear fraction of E549, that particular dose was showing most effective, most potent effect to decrease the NRF2 level. Then, though the KIF1 was mutated, its level increased with the particular dose, not only in the cytoplasmic, but also in the nuclear fraction. Then we wanted to study the subcellular localization, and I found that NRF2 was coming out of the nucleus, whereas the KIF1 was getting inside the nucleus. All these findings were supporting the hypothesis of KIF1 mediated nuclear export of NRF2. Then we directly went to the downregulating downstream molecules, and first we checked the activity of the enzymes NQ1 and SOD1 and found that there was only a slight decrease in the catalytic activity, but there was a significant decrease in the expression level of NQ1, then SOD1, and the drug transporter MRP1 was also not an exception. So with this, I want to summarize all my findings. Like, initially, black tea reduced the excessive oxidative load. As a result, the need of NRF2 got reduced. So, and black tea, not only that, black tea reduced the NRF2 level itself and also impaired its nuclear translocalization. On the other hand, it increased the KIF1 level, definitely not through by reverting back the mutation, but maybe through some transcriptional, post-transcriptional, or epigenetic modification. And it also encouraged KIF1 to drag out NRF2 from the nucleus. As a result, the downstream molecules like NQ1, SOD1, MRP1 was also downregulated. So we can conclude from our entire finding that in a less oxidative stressed condition where the need for NRF2 was also reduced, KIP1 was regaining its control over NRF2 in a better manner. As a result, the NRF2 pathway was more controlled, which was very similar to that of an intermittent activation of a normal cell. And I liked, without acknowledging my funding agency, that is Indian Council of Medical Research, and director of my institute, Chittaranjan National Cancer Institute, I can't finish my talk. And last but not the least, how can I forget all my lab mates and of course my mentor? Without their constant support and suggestion, I won't be able to do this work. Thank you.
Thank you, Ms. Shuchi Snigdha. So those who are drinking black tea, they can relax and continue drinking. The topic is open for discussion. It appears there are no more questions. So may I request Professor P.M. Gopinath to hand over a memento as a token of appreciation. Let us move to the last presentation of the session by Mr. Lohit Gowda. The title of the topic is Comparison of Anti-Cancer and Apoptotic Potential of Morus indica Varieties on HT29 Cell Line. Mr. Lohit Gowda is a research fellow pursuing his PhD at the University of Mysore. He has published in the international journals, and he has been awarded or felicitated with the Certificate of Excellence for winning the Best Oral Presentation Award for the paper entitled Effect of Nutrition Impact Symptoms on Quality of Life in Subjects with Head and Neck Cancer, Global Cancer Summit International Collaborative Conference, Institute of Science, Bangalore. So let me welcome Mr. Lohit Gowda for the presentation. You have 10 minutes. Thank you.
So good afternoon, everyone. Today, I'm here to present the research work entitled Comparison of Anti-Cancer and Apoptotic Potential of Morus indica Varieties on HT29 Cell Line. So, this research was carried out under the supervision of Dr. Asna Uruj. So, as we know, in recent years, medicine plants have attracted huge attention for their therapeutic properties and biological activity. So medicine plants are also used in various traditional and complementary and alternative systems. So especially for cancer treatment. So with this background, we aimed at investigating the anti-cancer and apoptotic potential of Morus indica varieties on ST29 cell line. So as we know, the Morus indica is a primary food plant for it, which is used for to feed silkworm in sericulture industries, and various phytochemicals are also being identified. Phytochemicals such as polyphenols, carotenoids, and vitamins A and A, C, E are also identified by previous researchers, and it is also being reported for its biological activities such as hypoglycemic, hypolipidemic, antioxidative, and anti-inflammatory properties. So in our laboratory, we have worked on Morus species for its anti-diabetic effect, anti-hyperlipidemic effect, and even antioxidant effect. So this prompted us to carry out anti-cancer to validate the anti-cancer effect on Morus species. So the methodology involves the procurement of V1 and G4 varieties, which is commercially available, and it was procured from Sericulture Department of University of Mysore. So the later it was extracted solvents using two solvents, namely aqueous and methanol, and the cell lines, HT29 cell lines were procured from ATCC, and later it was validated for its the extracts were validated for its cytotoxic effect using MTT assay. So based on these results, the extracts exhibiting a greater activity was then validated for its apoptotic effect using annexin 5 FITC assay against the SC29 cell lines. So in this table, you can see the cytotoxic effect of all the extracts. In this, V1 aqueous had shown a low IC50 value, and it was followed by G4 with 137.7 μg per ml concentration. So based on these results, we carried out apoptotic effect to validate the apoptotic effect using FITC assay by flow cytometry. And in this section, you can see in late apoptosis section, you can see the colchicine, which is in red color. It is a standard drug, and our extracts V1 and G4 had shown a similar activity. But here V1 we had used a lower concentration of 160 μg per ml, whereas G4 methanol extract we had used 320 μg per ml. So the major findings of this study, it was observed that V1 and G4 had shown a dose-dependent inhibition of growth on ST29 cells, and also the percentage of late apoptotic cells were found to be higher in V1 aqueous when compared to untreated cells, and also it showed a similar activity as colchicine. So the conclusion is Morus indica varieties, V1 aqueous exhibited a better potential than extracts of G4, and based on the results of cytotoxic and apoptotic effect, V1 aqueous proves to be a potential candidate for the usage of herbal medicine in the field of cancer, and because the results were comparable with the standard drug, and also it is essential to carry out further in vivo studies to validate its dosage and prove its potential as an anti-cancer agent. So I would like to acknowledge UGC and ICMR for funding this research, and also Skanda Life Science for providing the facilities. I would like to thank you, the organizers of EMSI conference, for providing me the opportunity to present in this conference. Thank you.
Thank you, Mr. Lohit Gowda. The topic is open for discussion.
I think